Good evening, everyone. So welcome to the webinar series by IITACB. So as we all know that the center, IIT Alumni Center, Bangalore, provides a unique networking platform to basically establish a collaboration between the academia and industries through the projects. So please be in touch if you have any specific projects in mind. So today's seminar is very interesting. Again, an interesting topic on role of plant microbiomes for food security. And the speaker is Dr. Shilpi Sharma, who is currently the chair professor at the Department of Biochemical Engineering and Biotechnology, IIT Delhi. It's brief about her. So she joined IIT Delhi in 2008 as an assistant professor. And before that, she worked as a lecturer in Motilal Nehru National Institute of Technology, Allahabad, and also completed her postdoctoral research at Helmholtz Zentrum, Munich. So Dr. Shilpi earned her PhD from the Ludwig Maximilian University, Munich, and she was gold medalist for her both the degrees of MSc and BSc at BHU, Varanasi. So again, her excellent journey as a researcher. She authored 110 journal publications and 20 book chapters and completed several sponsored projects. And her accolades are, she's currently the fellow of the National Academy of Sciences India that she received this year. And also she has received Tata Transformation Prize in Food Security in last year, 2023. And also she selected for the Women Leadership and STM program by Grand Challenges India and Women Lift Health this year. Among international recognitions, she is also the Mercator Fellow at the Technical University of Munich, and also she's a life member of New York Academy of Sciences. Yeah, so yes, so I think many more I can keep on going. And the talk will be moderated by Dr. Meenakshi Grover. So she is a principal scientist in the Division of Microbiology at the Indian Council of Agricultural Research, Indian Agricultural Research Institute. And her expertise is in microbiology. So mainly her research is focused on plant growth promoting rhizobacteria and stress resilience in crops and sustainable agriculture. And she is so the recognition she has received in terms of several awards, and which I'm just mentioning few of these. So she received the multidisciplinary team award from this institute, the Pearl Millets in 2018 and 2020. 2 years, and she serves as an associate editor for the World Journal of Microbiology and Biotechnology, Springer. And also she has received the Visiting Scientist Fellowship in USA, and also from the Department of Science and Technology, the Women Scientist Fellowship. So yes, so we look forward to this interesting talk, and today's webinar will be co-hosted by Dr. Sushila Venkatraman and myself. And for audience, so please post your questions or anything which you are curious about the topic. So that will be taken up during the discussion between the talk. So over to Dr. Sharma.
Thank you very much, Abha and Sushila, for giving me this opportunity. I'm just trying to share my PPT first so that we can start. So I hope my PPT is visible now.
Yes.
Also in the full screen mode?
Yes.
Okay. So a very good evening, everyone. Like Abha mentioned, I'm from the Department of Biochemical Engineering and Biotechnology from Indian Institute of Technology, Delhi. And I've titled my talk as Rooting for a Change Through Plant Microbiomes for Food Security. In a minute or so, I'll talk about the plant microbiome. And the way we've structured this talk is that I'll first give a brief in the first part of the talk about the topic and its significance. And then I'll delve deeper into the science that I do in terms of attaining food security. In 1937, the 32nd President of the United States emphasized on the need for effective soil management and wrote, "The nation that destroys its soil destroys itself." Still, the lifeline of Earth, that is the soil, has been largely ignored. Now, with a background in botany and in plant microbiology, I understood the might of the soil in determining plants' fitness right from the start. And I have since then been putting in efforts to restore this component. This is a picture which we're not really proud of. Indian population, we surpassed the Chinese population last year. So it's just showing the kind of pressure that Indian agriculture right now is facing. To add to that, the per capita arable land in India is way below the world average, and it's steadily declining in the last few decades. So Indian agriculture, together with facing the issue of increasing mouths to feed, is also experiencing deterioration in land due to climate change. Hence, the need for enhancing agricultural productivity comes with the need to also minimize chemical amendments and look for more environment-friendly alternatives. Now we're looking at the next green revolution, but here there are certain key terms or key aspects that we need to take into account. To attain food security, we're looking at environmental sustainability, biodiversity, plant health, and soil health. Now, when I say soil health, it's defined as the continued capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. And soil health has three major components: the physical properties, the chemical properties, and the biological properties. Me being a biologist focuses on the biological properties, which are impacted by both the physical and chemical properties, and the biological properties in turn affect these two. When you look at the fauna and the flora of the soil, you will see that the microorganisms, in fact, are the bottommost in this food pyramid that you see. They are not just abundant, billions in 1 gram of soil, they're also very diverse, and they have a major role to play in the nutrient cycling and the ecosystem. I wish to take a minute to clarify certain terms which have overlapping definitions. When you talk about microorganisms in a particular niche, and you mention microbiota, it refers to the sum total of all the microorganisms present in that system. So basically, we're looking at the taxonomy, what is present, how many of them. And on the other extreme of this arrow, you see functionality. So when you talk of metagenome, you're referring to the total DNA of the system, and studying that would give us an idea of the functionality or the total potential of the system in performing different functions. So in a simpler term, the microbiota can actually be looked as the musicians in an orchestra, and the metagenome it refers to all the music that the musicians can play or have the potential to play. However, when we talk of the term microbiome, we are referring to the orchestra which is being played by the musicians in view of all the music that they know. However, there's a very strong element of the environment influencing the kind of music that the orchestra is playing. And so, microbiome, when we talk of the term, it actually refers to microbiota plus the theater of activity around it, which is influenced by the environment. Now, when I say theater of activity, I'm referring to internal, external structural elements, microbial elements. It could be metabolites that are extracted. So the entire plethora of this activity and the microbiota is being referred to whenever I say microbiome. Now, I'm talking about the soil microbiome, which has been considered as a game changer for agriculture. Why? Because of the strong impact it has on various verticals. So, of course, the obvious thing is its impact on food security, because it is a matrix that supports plant growth. It is also a matrix which controls pests. It has a role to play in maintaining nutrient balance, in food safety, and also environmental sustainability. Now, if we harness the might of the soil microbiome, we can actually attain a number of SDGs. I've just listed 6 here, but there are several others which are indirectly also being affected by the soil microbiome. And in perfect analogy to the healthy gut and unhealthy gut. So for example, the unhealthy gut is believed to have dysbiosis or alterations in its microbiome, while the healthy gut is more diverse, it is more abundant and functionally more potent. We see that the soil microbiome serves as an excellent indicator of the health of an ecosystem. So while a healthy ecosystem is characterized by increased biodiversity, by enhanced soil functionality, we know that an unhealthy ecosystem has a compromised functionality because of alterations in the soil microbiome. The same principles that lead to the colonization of the gut and other parts of the human or the animal body stands true for soil microbiome and also the plant microbiome. In fact, the soil microbiome has been considered to be a connecting link in the One Health approach. The One Health approach actually says that the health of all individuals and the ecosystem are actually interconnected. Now, I'll take you through this figure just to make this clear. The soil microbiome is the source from which the plants recruit their microorganisms. And so is the case with the animals and human beings. They have a part of their microbiome recruited from the soil microbiome. In fact, that's how the gut microbiome is actually influenced by the soil microbiome. And the ingestion of the plant microbiome leads to a part of their microflora being transferred to these higher organisms. So there's actually a strong linkage between all these components, and soil microbiome happens to be a major component. So much so that we are now thinking of, or there are people proposing soil microbiome to be one of the verticals in the One Health approach. Now, I'm going to talk about a very specific zone, that is the rhizosphere. Now, as the name suggests, rhizosphere is the zone of soil that is surrounded, that is surrounding the roots and is directly influenced by the roots. Now, how is it influenced by the roots? The roots are actually secreting a range of different metabolites, and in turn makes this zone really rich. Rhizosphere is considered to be one of the major hotspots of microbial diversity and activity. And the way the rhizosphere is colonized by the microbiome, like I said, the source happens to be the soil from which the plant selects a particular community around itself in the roots. And a subpart of this actually also moves inside the plant parts, and this zone is called as the endosphere. Now, why is this important? Well, the rhizosphere microbiome has a very important role to play in determining the plant's fitness. So starting from acquiring nutrients for the plant, like N acquisition, it would help in P solubilization, in iron acquisition. It helps in mitigating different kinds of stresses. So for example, abiotic stresses like drought stress, salt stress, and various other stresses that we know of. It is also having a strong impact in suppressing plant pathogens. In fact, in current context, when we consider the plant and the higher organisms as a holobiont with very tight association with the microbiome, it is believed that the microbiomes having a shorter generation time can adapt to stresses much faster than the plant. So by the time the plant is adapting to a particular stress, the microbiome would anyway have adapted, and it confers selective advantage to the plant to adapt to the particular stress. Now, what drives the compositional assembly? Well, there are several factors related to the plant, which decides the recruitment of microorganisms. There are several factors also related to the soil, which contributes to this assembly. And in fact, there's everything in between. The plant microbe communication helps in recruiting the kind of microorganisms in the vicinity of the plant, and especially in the root zone. The idea is to be able to transform a non-optimized metaorganism or a holobiont, which has a compromised diversity and functionality, to an optimized metaorganism, which has enhanced NNP cycling, it has improved growth, enhanced disease suppression. And this could be a sustainable means. So this is basically what is called as engineering the rhizosphere. Now, this can be done primarily by three approaches. We could do plant-based engineering of the rhizosphere. When I say plant-based, we're looking at methods like developing transgenics or selecting cultivars or doing plant breeding, by which we can select plants that have a specific exudation profile, selecting the plant beneficial microbiome in its roots. When I say meta-organism-based engineering of the rhizosphere, I'm looking at the holobiont per se without delineating the two components of plant and microbiome. So approaches like co-engineering or crop rotation could be used. Now, these are tedious processes. We revert to the rather simpler approach of microbiome-based engineering. Conventionally, we have been using microbial inoculants, which have also been included under the strategy of microbiome-based engineering of the rhizosphere. Or we could also work towards looking at the microbiome in toto and drifting it or evolving it towards a plant beneficial one. Now, like I said, it's been decades that we've been using the might of the beneficial microbes as inoculants and bioformulations in agriculture. These are some of the common ones that I've listed here. However, we have realized that in the last few decades, they suffer from several challenges. The prime limitation is that the selection of a bioinoculant strain has been done or is usually done under controlled conditions. It is characterized under controlled conditions, and when taken to the field, there is inconsistency in its performance. Then the shelf life is a question. We are still not really clear of the larger picture of the mechanism of action of these bioinoculants or the beneficial microbes. Their action is slow. Their economics is compromised, especially when you compare them to the conventional inoculants. One of the prime reasons for these limitations, and especially the first point when I say inconsistency in performance in fields, is because they have to compete with the resident microflora, which has established itself in the natural conditions. So it's important that we actually understand the plant microbiome soil nexus and how the three actually regulate the assembly of the rhizosphere, which we've been doing for the first decade or the formative years of my group. We've been trying to really understand or untangle this nexus. Now, focusing on this talk and the topics that I'm going to talk about in the next part of my talk, I've listed some questions that I'll be dealing with. The first one being, can the efficiency and shelf life of conventional microbes be enhanced for increased competence under natural conditions? Then, can we use cell-free formulations instead of live cells? If there are so many limitations associated with the survival and the efficiency of live cells, can we really bypass this and use only the active ingredients for enhancing plant productivity? And finally, is it possible to manipulate the plant microbiome in situ without delineating the different components? Now, I'll try to address these questions in the second part of the talk, but I would like to invite before that Dr. Meenakshi for some questions or discussions if there are any.
Yeah, thank you very much, Dr. Shilpi. And like you have started really wonderfully with a very beautiful quote that the nation that destroy their soil, they will destroy themselves. And that is the reason nowadays a lot of focus on One Health concept, like worldwide, it has gained momentum like anything. So One Health, if we talk about the soil is the important component. If soil is healthy, we'll get a very like healthy produce. And if we consume better produce, we'll be healthy. So that is very beautiful linkage. And we all are like partners, stakeholders in this, being researchers and the farmers, all and even the young generation, they need to understand this One Health concept. So then we have moved towards like the traditional bioinoculants. What are the challenges associated with them? As you said, the rhizocompetence is a very important issue because particularly in case of India, because we have such a geographical diversity. So if we use one inoculant at one particular location, it may or may not perform at a different with slight different soil profile or the vegetation. So those kind of challenges, and then shelf life you definitely mentioned. So now if we come to microbiome, so how it will be advantageous, just for the sake of the students, if you could elaborate on this, how it can be advantageous, because in traditional inoculants also we have several challenges. So how this microbiome is going to be superior or better as compared to that part? So please.
Yeah, that's a very important point. Actually, uh, like I said, the major challenge with introducing a single inoculant or let's say a consortia happens to be the fact that um, you're sending a soldier, just a soldier, out to fight all these problems. And if I were to speak in very simple terms, when you're talking of microbiome applications, you're actually sending an army. There's a network of interactions which you try to maintain. Hence, this is considered to be a more robust approach than just individual inoculations, which needs to fight with the ongoing or the established interactions.
Yeah, that's very, very true. And another point an audience has made, like you, as you mentioned, that economics is a concern if we go towards microbiome. So can you elaborate a bit on that? And the other thing is we are emphasizing a lot more on natural farming these days.
Yes.
So what is your take on that?
Well, the point that I made on economics was not on microbiome, but on microbe-based formulations that we have right now. And that was especially in terms of comparison with the chemical fertilizers, which are much faster in terms of the effect that they exhibit. So that's how it is considered to be less economical compared to the chemical fertilizers. So, yeah, I mean, that's
Yeah.
what.
So yeah, that's very, very true. And natural farming again is again microbe based, if we say.
Yes,
So
yes,
the
right,
whole concept is revolving around the microbe because they are
right.
using the products which are fermented.
Yes,
So the natural indigenous microflora is like favored. So that
yes.
is how the natural farming is performing actually. So
So the concept is, yeah, I'm sorry. Please
yeah,
go ahead.
please go ahead.
No,
Please go ahead.
I was just saying that the whole idea is to be able to restore soil health and the
Yeah.
biological component here. So it could be means of amendment with compost. It could be means of amendment by different kinds of bioformulations. But ultimately, we are all looking at enhancing soil health.
Yeah, that is the main goal in sustainable agriculture actually. Productivity is one thing, quality is another thing, and the soil health and sustainability. What are we going to give to the next generations? So that is liability on us. So definitely we have to work in this direction. And you mentioned about the rhizosphere.
Yes.
So what are the different factors like that influence these plant microbes? They are very intimate, very intricate relationships. So
Yeah.
what are the different factors which influence these interactions?
As you ask me this question, it also leads me to the kind of challenges we face because of the plethora of factors which affect the rhizosphere recruitment of microorganisms. So it could be the edaphic factors, it could be the environmental factors, it could also be the plant. So from the same soil, if you grow two different plants, because of the rhizodeposition profile, they would select a very specific community around them. So this is
Exactly.
also one of the points that you mentioned as to why we cannot have a universal formulation, right? Because of all these factors impacting its colonization, impacting its efficacy.
Exactly.
So yeah, there are a plethora of factors.
And there are other things also. Once we isolate a strain, which is very efficient, but
Hmm.
over a period of storage times and all that, it start losing because it start adapting to those cultured conditions. So that is again a challenge. So we have
Right,
to work more on in situ type of strategies, I think.
right.
So let us check if there is any other question. So otherwise, we can move forward. So I think let's have like about what is your research and some in-depth information. Then we can continue again.
Okay, so if there are no further questions, and if I have been able to communicate the fact that the microbiome and its might can be harnessed for enhancing crop fitness, I would like to now talk about my contributions to addressing this, my bit actually in contributing to in addressing the issues of food security and agricultural sustainability. The goal of my working group is that in view of the issues that we have just outlined, the climate change leading to soil degradation and the increasing population, we are trying to look at microbiome-based solutions for food security and sustainability. That is the underlying goal for all our work. With the plant microbiomes having shown their might as drivers of the next green revolution, we are trying to harness this through various projects. I've listed down some of the projects wherein we are working towards. Just give me a second. I think it is. Yeah. So the first question that we're trying to see is whether we can enhance the efficiency of the conventional bioformulations in view of the methodological and technological advancements that we've had. Can we develop next generation bioformulations? Another ongoing project is on plant-mediated in situ microbiome manipulation. And actually, this is an extension of what Dr. Meenakshi just now mentioned, that we're now looking at alterations or engineering in situ so that we can surpass the major challenge of the colonization of these artificially introduced microorganisms. The third project that we're working on is trying to devise a farmer-friendly strategy of identifying the good soil and then applying them to transform the bad soil. So this is called as rhizomicrobiome transplantation. And finally, we've also ventured into using satellite-based hyperspectral imaging for assessment of soil and plant health, and then the manipulation. Now, I'll take up two stories in my talk in the rest of the time that has been assigned to me. However, I briefly want to touch upon these two important aspects as well. So I have just one slide on each. What do I mean by rhizomicrobiome transplantation and what we do here? Well, this is the one which is actually sponsored by Tata. They are the ones funding it. Tata Sons. What we do in this is we try to identify the good soil. When I say good soil, I'm referring to the soil which has very high biodiversity. It is also the one which has increased functionality. It shows suppression to different kinds of pathogens. And we then characterize the microbiome and strategically transplant this to conducive soil or soil which is not good, trying to transform this. And in this initiative, what we've done is we have kind of mapped several states in the country with the objective of identifying soils which are naturally suppressive to a range of pathogens, and then using this soil to transform conducive soil, wherein you have a lot of manifestation of plant pathogens, by using this farmer-friendly strategy, by simply mixing the good soil and the bad soil. In case of hyperspectral imaging for assessment of soil and plant health, what we try to do is we're focusing on using satellite-based hyperspectral imaging for assessment of not just plant health, but soil health. And here, what we've started off with is trying to use hyperspectral imaging for early detection of plant diseases. So, for example, we're working on wilts in pigeon pea, and we developed our models from an experimental field in Uttar Pradesh, and then moved on to validation of this model in fields in Gujarat. So this is the kind of spectral images that we generate, and we try to see if there is a detection which is possible, and you can understand the strength of this technique in being non-invasive and several other advantages. And we have recently published this nice report on early detection of wilt in pigeon pea, wherein we were in fact able to detect wilt much before even the multispectral imaging data that we could get. With that brief on these two projects, I would like to now move to these two other projects in a little more detail. The plant systems that we work in the group include two legumes, mung bean and pigeon pea, and we also work with tomato and wheat. The basic approach is what has been summarized here. We try to adopt both the reductionist approach and the holistic approach. Now, what I mean by the reductionist approach, which is more bottom-up, is that we try to characterize the beneficial bacteria by culturing them, trying to isolate them. We characterize them in terms of their ability to promote plant growth, their ability to suppress different plant pathogens. And then we start making bioformulations by either using the single isolates, we try biostacking them, or even generating consortia. Now, this is the more conventional approach. However, I'll talk about how we try to amend these approaches for enhanced survival. The holistic approach, on the other hand, it is more top-down, wherein we try to identify the beneficial or the microbiomes which show enhanced plant fitness or enhanced plant protection. And then without really dissecting the different components, without attempting to culture them, we try to go for application of these microbiomes per se. We also generate synthetic microbial communities in this approach. And the two questions that I'm going to deal with are one each from these approaches. I'll talk about how we have been trying to enhance the survival and efficiency of conventional bioformulations. And I'll also talk about in the second part of the talk, if plant-associated microbiomes can be modulated in toto to confer targeted benefits to the plant. Starting with the first one. In developing next generation bioformulations, what we're trying to do is we're trying to design bioformulations which are metabolite based with the idea that they would exhibit enhanced survival under the field conditions. We're also trying to optimize the really conventional additives. We're also looking at the choice of alternate carriers and bioformulation types, especially the economical ones. I will restrict to the first point, that is metabolite-based bioformulations. Now, conventionally, what we do is we cultivate microorganisms which are identified as being beneficial to the plants by conferring various properties. We formulate them either in the form of liquid formulation as slurries or as solid formulations, and then we apply these either as seed treatment or as soil amendment or even as foliar spray. Now, the major challenge is at all these steps, you will see that there is desiccation. There's desiccation in soil, there's desiccation in foliage, and hence the efficiency, survival and the efficiency of the bioinoculants is compromised. This story is going to be on pigeon pea, which is the second most important pulse crop of India, and India actually contributes 90% of world's acreage and production. However, the productivity of pigeon pea has stagnated for the last few years. We have seen that it's stagnated. Another observation has been, and especially in the Indo-Gangetic plain, we see that there is very poor nodulation in pigeon pea. And in fact, this is a catch-22 situation. So because there is poor nodulations, farmers apply a lot of chemicals. And because there's a lot of chemicals which is being applied, the nodulation is poor. So it's kind of a catch-22. The productivity of pigeon pea is also affected by various kinds of stresses. This includes attack by the pathogen Fusarium udum, which causes the wilt. The bioinoculant we selected is a conventional Bradyrhizobium species which exhibits multiple traits. So it actually is an EPS producer, an exopolysaccharide producer. It produces plant hormones like indolestic acid. It produces the iron chelating agent siderophores, a chitinase producer, and it's also a phosphate solubilizer. Now, what we noticed is that this particular strain is also showing biocontrol activity against this major pathogen in pigeon pea, that is Fusarium. We could see that it's actually affecting the mycelial structure. So basically, this strain was not just exhibiting plant growth promoting properties, but also biocontrol properties. So we considered this to be an ideal candidate to work with. We characterized the cell-free culture supernatant of this particular strain, and we found that there were a number of benzoic acid derivatives. There were a lot of amino acids and derivatives of coumarin, which gives us the first idea that the cell-free culture supernatant also has a lot of active metabolites which can be harnessed. We went ahead and compared the typical cell-based Bradyrhizobium formulations that are conventionally used by farmers. And we developed formulations based on metabolites. In this case, we use exopolysaccharides. We also used the cell-free culture supernatant, which I just showed you had an active profile of all these metabolites. We generated these bioformulations. And in typical flow of experiments for us, we would first take these treatments to the plant growth chamber. We would then go to the pot experiment and then perform experiments first in experimental field and then the farmer's field. I'm going to show you data on each of these sequentially, but some representative data for each. This is the result that we found when we use these formulations under controlled conditions against the pathogen Fusarium. F here stands for the Fusarium pathogen. B stands for the conventional bacterial cells which were applied. S is the supernatant and E is the exopolysaccharides. And you will see that even the supernatant, just the supernatant, was showing a remarkable decrease in the incidence of disease. And the effect was very prominent, positively prominent, in the case of the combinational bioformulation, wherein the bacterial cells, the supernatant, and the exopolysaccharides were applied. So this was one validation of the strain's ability to control the disease under these treatments. We then went to the experimental field, and this was conducted in the fields of IRI, wherein we tried to see if these amendments or these formulations could impact the resident rhizobia which is present in the soil. And again, C is the control, B is the bacterial cell, S the supernatant, E is the exopolysaccharide. And we could see that the numbers of the resident rhizobia were remarkably enhanced in these amendments, even in the supernatants. So this is how the things look like when we looked at just with the naked eye, when we looked at the combination bioformulation, the control bioformulation, which had no amendment, and the monoinoculation, which is the typical conventional Bradyrhizobium species, which is applied. Now coming to some numbers. Here I'm showing you the nodule number, which is an important aspect. I told you this is one of the limitations that we have in the growth of Cajanus cajan. And I'm also showing you one of the most important parameter for the farmers. We could see an increased nodulation in the case of these amendments, which was much higher than just the monoinoculation. And the seed yield was also, we observed a remarkable enhancement in the seed yield in all these treatments that we had. So the next question was, with experimental fields being, while they're still close to the farmer's field, they're still under a lot of intervention by the scientists. So we then moved on to a farmer's field, and we identified a farmer's field in Kanpur Dehat location, which was actually experiencing a number of stresses simultaneously. So for example, it had a pH, a very high pH, and it was the temperature in this region was very high. Farmers were applying 100% of recommended dose of chemical fertilizers. So we selected this particular field. And here, when we applied these formulations, we could see that. So for example, if you look at the control wherein there was no amendment, there was no nodules formed. And the same was the case when the chemical fertilizers were applied. However, in the case of application of these monoinoculants and the EPS-based formulations as well as the supernatants, we could really see an enhancement in the nodule number. And the seed yield, again, the result that we got in the experimental field could be well validated in the case of the farmer's field as well. So to summarize the first part of the talk, the seeds receiving combinational bioformulation, they helped maintain rhizobial population at pre-flowering and flowering stages. We saw that the combinational bioformulation was the best in terms of enhancement of plant attributes and also in controlling disease incidence. So basically, here it was serving, the Bradyrhizobial species was serving several features like controlling plant disease, like enhancing plant attributes, including enhancement of nodulation. We could identify various antimicrobial metabolites from the cell-free supernatant and zones of inhibition. So we were actually able to bypass the use of these living organisms, and the supernatant was found to be equally effective. And so the combinational bioformulations having diverse constituents could serve as next generation bioformulations. Now, after having talked about this very traditional approach of using bioinoculants, and after having shown that the efficiency can be enhanced not just under very controlled condition, but also under field conditions, we were interested in seeing if the microbiomes can be modulated without, like I said, without teasing them apart. So the question was, can plant-associated microbiomes be modulated in toto to confer targeted benefits to plant? Under this story, the kind of projects that we work on include, number one, we're working with various kinds of hosts on trying to acclimatize the rhizosphere microbiome under repeated cycles of stress. So the idea is we actually, the stress that we apply onto a plant is with the idea that the plant would select its own rhizospheric microbiome. And so this will be a more robust approach than applying something from external. So this is more in situ modulation. Then, like I mentioned, we also work on rhizomicrobiome transplantation, the story that I just mentioned to you. And we're looking at several amendments which can modulate the microbiome for the better. Now, in this part of the talk, I will talk about the first story, which is acclimatization of the rhizosphere microbiome under repeated cycles of stress. Here, the model plant is Vigna radiata, and I'm talking about stress, which is salinity stress in this case. And I would like to elaborate on this methodology, which is novel in terms of acclimatizing the rhizosphere microbiome. Now, in this case, what we do is we perform community coalescence. By community coalescence, I mean to say that we try to bring together microbial communities from different sources just to give or prepare a starter inoculum which has very high diversity of microorganisms. And then in this coalesced community, we grow our plant under stress. Now, what happens is the plant selects a microbial community which would help it mitigate the stress. From the pool of plants that we have, we select the one which is the best performing one in terms of its attributes. So whether it is the phenotypic attributes like plant height, the shoot length, root length, we try to select the best plant. And what we do is we use the rhizosphere soil from this best performing plant as an inoculum for the next plant growth cycle, wherein this inoculum is applied. And this is also done under stress. So basically, we are exposing the plant to repeated cycles of stress. And in every passaging, we're taking the rhizosphere microbiome from the best performing plant, using it as an inoculum onto the next cycle. Eventually, after these multiple rounds, what happens is we have the microbiome, which is evolving faster than the plant and helping the plant mitigate these stresses. So we consider the microbiome to be acclimatized when the controls show the same parameters as the application of the acclimatized rhizosphere microbiome. Now, what is actually happening here? When we are exposing the plant holobiont to repeated cycles of stress, there is alteration in the indigenous microbiome. And this is a microbiome, please remember, this is a microbiome which the plant has selected on its own. So what happens is the stress would lead to the selection of stress-tolerant microbiome. And the other strategy of the alteration is that the plant's exudation profile also is altered, which then selects an altered microbial community around itself. So these are the two forces by which we are evolving the microbiome for benefit to the plant under the particular stress. Now, what did we do in this case? In this story, we started off, like I said, with Vigna radiata under salinity stress and tried to acclimatize the rhizosphere cycle over repeated microbiome over repeated cycles. We then were also interested in seeing how the bacterial and the fungal community evolves along the passaging. So we tried to characterize the bacterial and fungal community. The marker for us to understand whether or not the acclimatized microbiome is effective in enhancing the plant fitness, that was done by quantifying various stress markers and various plant attributes. A major challenge in microbiome engineering or using rhizosphere engineering in large-scale application is the fact that people across the globe are working towards developing methods for storing the microbiome in the matrix to retain its functionality. Now, in the absence of this particular method, if we have an acclimatized microbiome, the only way out was actually to generate a culture bank of potent salt-tolerant strains, characterizing them for plant growth promoting properties, and also trying to understand the mechanism of action by which it is mitigating the stresses. And once we had this robust culture bank in hand, we could easily take them to the field application by generating syncoms. Strategically, we designed syncoms for stress mitigation and then tested their efficiency in planta. Like I said, the sequence is always control condition, then moving on to experimental field and the farmer's field. Now, what did we observe? This is just a representation of the kind of impact that you could really see by application of the microbiome. What I'm showing you here is plants in the 13th passaging cycle. We went on actually to do these passaging cycles for about 23 or 24 plant growth cycles. And the ones in M are actually referring to the microbiome applications. S here refers to the salinity stress treatment, while P is the control plant. When we looked at the plant parameters, and I don't want to really confuse you with this, I want to draw your attention to the last 3 bars, if you see, the yellow, blue, and green bars. As we moved on, what I'm showing you here is from the 7th cycle to the 13th cycle, you will see that all the treatments with the microbiome application showed incredibly higher values compared to the other passaging. So basically what it indicated was that the microbiome was getting acclimatized and it was helping plants mitigate the stresses. Now, this was in terms of the dry weight. We also, like I mentioned, looked at several other indicators. So, for example, the MDA level is one indicator of the stress that the plant is exhibiting. The membrane stability index is another indicator of plant health. And using all these parameters and much more, we could actually see that the microbiome was conferring an advantage to the plant under stress. Now, what was happening to the microbiome? We studied the bacterial and the fungal component. What I'm trying to show you here is just the bacterial component to show you that as the passaging was performed, and here the subscript is referring to the number of generations. So for example, M5 is the 5th passaging cycle, M13 would mean the 13th passaging cycle. As we progressed, we see the selection of a very specific community under the stresses. And looking at some of these diversity indices, we saw that the treatments under stress with the microbiome were actually enhancing the rhizospheric microbial diversity, which was the objective of this entire experiment. Now, like I said, we had to revert to cultivating and generating plant growth promoting microorganisms from this acclimatized microbiome because the microbiome could not be stored for later use. So we tried to form this robust culture bank. We characterized all these strains that we had. We checked their efficiency and the performance under different kinds of different levels of salinity stress. And then the first experiment that we wanted to perform was, if we have this robust culture bank, how effective are these individual strains in enhancing plant growth and in also mitigating the stresses. So what I'm showing you here is just 4 different strains, wherein the C is actually referring to the individual strain without the stress, while D refers to, in all these panels, the individual strain together with the salinity stress. And this gave us the first indication that even the individual strains were very efficient in this process of enhancing plant fitness. So this gave us the confidence to then move on forward. For their performance in the field conditions, we then wanted to actually bring together strains to form this robust amendments, which would actually be competent enough under the field conditions. So we generated several syncoms, and our idea was to see if the syncoms were at par with the total microbiome. And in fact, all these plant parameters that we tested, the dry weight, the stress indicators like the membrane stability, the number of pods, they all showed that the syncoms that we had generated were equally efficient to the microbiomes. So they could be taken forward for large-scale application. These are some more of these parameters. So for example, the chlorophyll content or the proline content, the malondialdehyde levels, these all showed an enhancement over the control plants wherein the microbiome application was not made. So the big question then was, we again wanted to see if these methods could be validated under natural conditions or farmers' field. And here again, we had a field in Uttar Pradesh, which we identified. And when we applied our microbiome, this is the kind of impact that we had. So this was a field which was left barren by farmers because of the very high content of salinity. And this is the picture that we got after we amended our microbiomes and the syncoms. Without really going into the numbers here, I would like to summarize this part of the talk. This is by far the first study to employ multi-passaging of rhizosphere microbiome of Vigna radiata, which was generated under stress, under salinity stress. And in fact, as I speak, we're trying to also ramp up the level of salinity here to see if the microbiome can be adapted to really high values of salinity. The stress acclimatized microbiome application led to an enhanced plant growth not just plant growth attribute, but also reduced stress marker levels. We could track the dynamics of bacterial and fungal communities which were being evolved during the process of passaging, and we could identify the stress tolerant members. We were then able to generate this robust culture bank that we have. From this culture bank, we tried to design SMCs, and we saw that the synthetic microbial communities or SynComs enhanced plant growth attributes. So basically, this acclimatized microbiome could be taken into the field. This was possible both under control conditions and under the field conditions. And the microbiome-based rhizosphere engineering thus has shown its efficiency in mitigating stresses as a potent tool. What next? Now, after these success stories, there's several questions that have actually opened up, and we're working on these. The first question is, can the microbiome members be tracked? We are not looking at delineating the microbial components. And then what is the strategy of then being sure that the microbial members have been competent enough in the field conditions? What is the spectrum of hosts for the engineered rhizosphere microbiome in mitigating stresses? We have generated an acclimatized microbiome for salinity stress for Vigna radiata. And I mentioned to you that there's several factors, including the plant genotype, which determines the kind of microbiome that can be colonized. So is this acclimatized microbiome going to be efficient? How efficient is this for different kinds of hosts? We're also testing the efficiency of this microbiome for different kinds of stresses. So right now we're working on drought stress as well. What are the active ingredients responsible for their impact on the plant? Is it possible to delineate the metabolites, and then is it possible to then go cell-free once we have generated the microbiome and understood the mechanism of action? And then the big question, how practical is the field application of microbiome-based rhizosphere engineering? The major bottleneck right now that we're facing is, like I mentioned, the lack of a method of storage of these soil microbiomes to retain its functionality. And once we cross that, the other major question is, how are we then going to propagate this microbiome, the acclimatized or the good microbiome, for application in field conditions? With that, I would like to thank, there are a number of super collaborators that I've had. I've listed only the ones which I'm collaborating right now with. I'm collaborating with people across the institute, from my department, from the Department of Mathematics, Electrical, Civil, and Center for Rural Development. There are a lot of collaborators that I need to thank, also across India and abroad. There are several funding agencies who have generously funded the stories that I've been talking about. Most of all, I would like to acknowledge the efforts of my team. This is, again, very dynamic. It changes every 6 months. So I've tried to put 2 pictures which have kind of covered the stories that I've mentioned. Shubham Dubey is my PhD student who worked on the mitigation aspect. And the Sonal, Rashi, Salila, they're also working on acclimatization. That will be all from my side. Meenakshi, I'm open to questions.
Yeah, thank you, Dr. Shilpi, for again a wonderful session. Your own research experience, whatever work is going on in your lab, and I like accolades for your team, huge team, and a good piece of work, and the divergence of like the research, like different aspects you are touching, the hyperspectral imaging also. That's very interesting to know. And it's these are the things of like present and future. So I really appreciate your experience and your sharing your research experience with all of us. So in your first piece of work where you are dealing with Cagenes cagen, so there we are targeting a fungal pathogen Fusarium mutum, right? And we are using a combination of the culture is cell-free supernatant as well as the metabolite. So when we are putting all these together, so could you study some of the mechanism, how these things are working actually when they are like with the plant system? So some mechanism, if you could elaborate or highlight for the students and for our own knowledge.
Yeah. So while I did not show that, I mean, due to paucity of time, we are actually trying to now see if we could identify these individual metabolites, use these individual metabolites, and see if those can lead to the same kind of impact that we've had. Because that will make our life very simple when we know the targeted metabolites. So that's one thing. So we are doing this. We're also doing certain assays by which we're trying to grow the pathogen and the plant together with the particular strain of interest, and trying to see what are characterize the entire profile upon challenging the pathogen with the pathogen to the plant and without. So we're trying to really identify these metabolites.
Yeah, fine. And another interesting thing with this metabolites is like we have to know their like the half lifetime in the soil and how they behave and how they interact, whether they are able to actually work function in when applied in the soil. So those things also we need to, like, I think, do in-depth studies,
Yes,
because the
and
moly
also
these
the
are,
dosage. Yeah, it's also
yeah,
important.
those, that's important thing I was about to mention. So could you see that a dose dependent effect also, or just a standard dose you have?
No, so definitely the dose is important. What I also
Yeah.
want to add is that the frequency of dosage and the
Hmm.
concentration, both of them are important,
Yes.
right? So there are a
Both
lot
are
of things
important.
that need to be standardized when we take. It's not as simple as just identifying a metabolite and applying it to the soil under
Yeah.
conditions. There's several other parameters that need to be established.
Exactly.
Yeah.
Yeah, and when we apply these things, definitely they are going to impact the indigenous microbial community also. That how they are influencing the indigenous microflora. That
Right.
also is a very important thing to take care of,
Right. So
right?
we call them,
And
we call those as the non-target effects.
exactly.
That's one vertical that we very strongly work on.
Yes, yes, that's very important. Okay. So could you elaborate little bit on what are these cell-free supernatant? So for the added information for the students, because many of the students, they might not be from the biology background, so it's very
Yeah, yeah.
important to know.
So what we do is we simply speaking, we grow these microorganisms in their typical culture medium. And after an incubation period, we try to just remove the cells so that we can bypass the limitation of the cell's survival in the control condition. What we have is then just the metabolites being secreted by the bacterial cell. And we call them as cell-free culture supernatant.
Yeah, and that's why the recurrent application of these
Yes.
metabolites comes into the picture, because live cells are not there in this case,
Right.
only their metabolites are there, so we may need to apply it at different like intervals, so dose
Yes.
and frequency of, like treatment also matters a lot here. Yeah, very very correct. And then you mentioned about this use of ready rhizobium plus these EPS based formulations and the cell free supernatants. So, can these formulation be extended to some other crops or some other like pathogen? Because
Yeah.
one good question is regarding phytophthora in
Yeah.
case of like horticulture crops, like avocado and all that. So, uh, quite definitely there is possibility, but yes, we need to. So,
Yeah.
what is your say?
So we have actually gone ahead and tried using some of the other conventional bioinoculants. So for example, we work a lot on bacillus species because of all the
Yeah.
advantages it exhibits. We have been working on bacillus species. Some of the newer ventures in this aspect is we're trying to use the EPS as the mobilizing agent, the immobilizing agent as well.
Yes.
So,
Yes.
but these are things in progress. So, yeah, yes, this point is well taken. We are extending it to other conventional. I mean, the acceptability is much more if you work with this conventional strain. So, we're trying to enhance
Exactly.
this. Yeah, the
Exactly. And definitely we can go and we can give trial in different crops with different pathogen these agents, pathogenic agents. So definitely there is a scope, and we may need to work on niche specific communities also. So if we are talking about horticulture crops, maybe we need to like isolate microbes from their those habitats. So for better functionality and all that.
right.
So that's fine. And there is another question, like we are talking about biotic stress in the first case. And in the second case, we have already taken already salinity as a example of abiotic stress. Definitely, we can work on other abiotic stresses also. So there is a point made by an audience, like, can we do such kind of work, microbiome-based strategies for heavy metal remediation? So, any experience or information can you share?
Yes, so this can be extended. I mean, this was just an example to show that these approaches work, and they are much better than using the single inoculants. Worldwide, people are trying to harness the microbiome now, and I know I cannot specifically point out to groups right now, but I know that heavy metal mitigation is also being worked very heavily, very strongly in different parts of the group. In my group, we restrict, as of now, only to salinity, drought, and phytopathogens. But yes, this can be extended to different kinds of
Yeah,
stresses.
exactly. Yeah. And I want to add here, you are working on EPS. In our lab also, we have initiated work on EPS. This EPS is a very wonderful molecule.
Yes.
So for heavy metal remediation also, EPS can work very well, and there are literature reports, many
Right.
reports are available, and people are working on microbiome based strategies for the heavy metal remediation in the soil also. So that is a very well taken aspect already, labs are working on that. So,
Yes.
and the other thing we are talking about, the acclimatization of the microbiome, a very
Hmm.
beautiful passaging you have explained. So again the point comes, as you also mentioned, how to maintain. Once we acclimatize this microbiome, we have a bunch of soil, right? So how to not lose the diversity which we are having in our hand? So that is a very important point. So what are the things we can do in this direction?
Hmm. Hmm.
Please highlight.
So, like I mentioned, this is a more futuristic approach. We
Yeah.
are yet to come over several bottlenecks, one of them being the preservation of microbiomes. There are several biobanks which are aggressively working towards developing a method for this microbiome. And once the jackpot is hit, I mean, this microbiome-based approaches are going to just it'll be a major transformation. But
Yes.
as of now, because we don't have a method in place, and there's several questions in place as to how long does this microbiome retain its ability, how long can
Exactly.
it, whether it can revert if it is not stored under the selection pressure, is it possible that it reverts to its original? Several
Yes.
questions surrounding this. I mean, I really did
Yes.
not go into all these details. However, the
Mm-hmm.
only practical solution as of now that we see is to develop a culture bank. Till we have a
Yeah.
method in place, you have to go back to the roots, get the culture
Yeah.
bank, and then try to
Definitely.
develop a more complex, I mean, move ahead from the consortia based approach, move a little forward in developing syncoms by understanding what are the interactions. Using some amplicon based tools, we can actually understand these interactions, generate the syncom based on this information. So sequencing assisted culturomics is what we're looking at right now.
Exactly. Very good point you have taken, sequence assisted culturomics. So
Yes.
this is the beauty of metagenome work nowadays. We get to know the vast number of diversity and the genera species which are present in the soil and in the environment. So the thing is, how can we target a few of them? We can make them culturable, because now we can get that functional net from this microbial community. We can get the network studies. So
Yes.
based on these, we can develop some strategies, some sort of media amendments we can do, so to make more number of unculturable microbes into culturable ones, and it's very easy to store and maintain them. Because
Yes.
for this information of the students, I want to add here, less than 1% of the microbes are culturable. Even today we say less than 1%. So
Yes.
it's a huge diversity. We are taking benefits because they are already working for us in the soil. They are giving all the benefits to plants which we are depending upon, but we are not able to culture them. We are not able to bring them into the petri plate or slant form. So that is very important, and that culturomic approach, a lot of focus is emphasis is being given nowadays on that part. So that is again a very interesting field for the upcoming new generation students. And for your information, Dr. Shilpi, I want to add, many of the students are interested in working with you. I'm getting like messages. So how to
Okay.
join your team? What are the options? So that is on a different way. They can contact definitely if there is something they can contact you personally. But just like this is the impact of your work.
Thank you.
So really like yeah, they are showing good interest. And the other thing like the storage we have already taken care of. And the microbiome transplantation, I want to add one more thing here. Like the fecal transplantation is really happening nowadays. We are seeing in India also. So a lot of cases, people are benefiting because years of like pain and we have that gastrointestinal problems people were facing. So that is working wonders because last month I think I saw a paper cutting or some report. So that thing is happening in India. And recently some report has come that people are working on brain microbiome also. So earlier it was thought that brain is sterile. It is
Right.
separated from the blood, this thing, circulatory system. But now it has been found that even brain is having a lot of its own microbiome, which can be related to the different neurodegenerative diseases. And maybe near future we'll be getting nutraceuticals, microbe-based nutraceuticals for the such type of therapies also. So that's again a good news. We are nothing without microbes. That is like very a clear picture now. And it is sad that there is no eukaryotic system without a prokaryote as its close associated entity. So that is a thing very very interesting. And now it's very important to make the youngsters aware how to take care of their microbial health. Very very important because the food habits are changing. So we are disturbing our microflora. So that thing we have to in the animal system also we have to take. And plant system like already like a lot of work, people like you, we are also trying. So that's a very good thing. So I think let us see if I have any other question. Yeah, the another important thing is race, like here like because we have again the diversity in Indian geographic. So a lot of factors are influencing, and we are talking about second green revolution. So even the traditional bioinoculants, the farmers are not much aware. In the interior India, only the program nowadays awareness is there. Definitely, there is increase in the number of farmers which are utilizing this bioinoculant. They are demanding, they are asking, but still there is lot more population which need to be like approached and make aware. Because if we go interior, because we have a mgmg program, mera gaon mera gaurav program, very like we are covering all nearby well like states. So our teams are going. So there we see that many farmers they do not know about these bioinoculants. So that is very like we sometimes say okay, we are doing work, we are organizing melas, kisan melas and all that, but still there is population which need to be approached. And how to make these farmers aware about this? We are talking about second green revolution. So what can we do about in that direction?
A very pertinent point, Dr. Meenakshi. I mean, whatever we do in this lab, it has to reach the masses, and the stakeholders happen to be the farmers. And in fact, as you asked this, I and my team, we are in the process of actually conducting what we call as need assessment.
Okay.
We're going to the farmers. And in fact, when I showed you this map of India, wherein we have been trying to sample from all these states, we also have a form in place where we're trying to gather data as to how many of them know about it and how many of them are accepting enough. So, because
Yeah,
acceptability is also one question. So,
exactly.
yeah, I totally agree that there has to be rigorous initiatives to actually go to the masses, go down to the stakeholders, to the fields. There are a lot of initiatives by the government as well.
Yes,
Just that, and especially the ICR system, I have to acknowledge
exactly.
the way that they interact with the farmers. I'm also very heavily relying on the ICR systems to take my work to the masses. So yeah, a pertinent point. I just hope people in this seminar listening to the talk are spreading this also to the actual stakeholders.
Exactly. It's like just the word of mouth, what we say, right? So it has to reach the information, that awareness has to reach farmers. If any student is from village background, he should tell his parents, his neighbors, like that. These things can spread. Yes, there is benefit. And what I generally, when we interact with the farmers, I just used to tell, if by spending 50 rupees you can save 25% of your urea, why not?
Yes. True.
And if there is no effect, you just come and tell us.
Correct.
Maybe
Correct.
the strain, sometimes it happens because of the quality, the products which are available in the market, they may not be very, like, quality may be issue sometimes. But if they purchase, if they get the product from, like, very reliable sources, definitely it works. And that's why we ICR system conducts a lot of frontline demonstrations to tell the farmers about their technologies. So efforts are in place, definitely, and we are hoping for a, like,
I think
good
you also raised a very important question, Dr. Meenakshi, and that is about the quality of the bioinoculants. This is one prime reason why farmers are losing confidence in the product, because we don't have quality controls. And I think I remember reading a paper on Indian bioformulations, and it's a pity that more than 50% of these bioinoculants actually had no live cells. So they were being, so quality control, I think there needs to be more,
Yes, it has to be rigorous. Now government is taking like steps towards that also, and they are generating centers for that quality testing of the commercial products also. So it will take some time, but I hope, because if a company has to sell a product, quality is the important thing. If it is quality product, it will definitely go. So if a farmer sees the fact, like, visually, and the thing is, in case of bioinoculant, the effect is not that remarkable in first, like, visual. But if we see very closely, we can point out, there can be difference in the greenness, there can be difference in the girth, so, and yield, definitely. So these things, if a farmer get to see, definitely he's going to believe, and
it
it
is
will
also
work.
important that we handhold the farmers, I feel. I mean, this has been my objective also, that once we handhold them, I mean, just saying it works doesn't really help. So really need to take them forward, hold them, and show them the impact, and then the word of mouth things spreads.
Yes. So there was another point about the endophytes. Would you like to throw some light about the endophytes and how, whether they can be used? Definitely they can be used. So little bit elaboration from your side will help, definitely.
So well, endophytes are also being heavily worked, not as much as the rhizosphere, just because of the logistic convenience, I would say. But endophytes there's some very potent strains, endophytic strains, and I know of really good groups working even in India in taking the endophytes to translation. There's several challenges also in terms of using the endophytes. The prime one is being establishment of the fact that it's an endophyte and it is going inside the plant is one challenge that when I talk to my friends who are working on endophytes would tell me. But yes, there's a lot of scope that I see even with the endophytes in taking them for not just plant growth promotion but also mitigation of stresses. So, Dr. Meenakshi, I think you also have some experience with endophytes.
Yes. So currently we are also working on endophytes, like from the pearl millet seed endophytic microflora we have explored, and we are getting really good functional microorganisms, like a diversity of functional traits, starting from the PGP, which we call plant growth promoting traits, like phytohormones, exopolysaccharides, then their ability to inhibit the phytopathogens, plus the lytic enzymes, an array of enzymes they produce. So they are remarkable. So we need to work in this direction to select the potentials one. Definitely they are the one which are closely associated when the plant is germinating. So they are the one to interact, and maybe they are helping in the plant nutrition also, or maybe protecting the plant from the surrounding harsh conditions. So there are there may be different modes of action. So yes, endophytes are they are really they work, and they are being explored for secondary metabolites also. An array of they are known to produce diverse types of metabolites which are of agricultural as well as industrial applications. So yes, for that question, we can say a big yes.
It is a
So
big-
I think if time permits, we can hand over to Dr. Sushila now. Thank you, Dr. Shilpi, for a wonderful interaction and session. Thank you so much.
Thanks a lot for moderating.
Thank you very much. A few years ago, if we would never have imagined such a talk by someone out of an IIT. So it is wonderful. From our point of view at the IIT Alumni Center, we've been running these webinars, 80 of them done, this is the 81st. We've, one of the areas we've picked up is the whole idea of Atmanirbhar Bharat. And we've looked at many, many areas around that. We've looked at manufacturing, we've looked at various technologies, we've looked at education and research. But food safety, which is the absolute imperative for us to be able to feed our billions, and we are the most populous nation in the world without the appropriate ratio of arable land. Given that whole thing, this one topic was something which we were missing in our story of Atmanirbhar Bharat. While I know there'll be many others to come in there, this was definitely one that was missing. So thank you ever so much for helping put that segment in for us. The next green revolution, you said, will require us to improve agricultural productivity, but do that in an environmentally sustainable and also in a health-friendly way, because the sicker we get, the worse the nation is going to get. And in a nation like ours, we can't really afford sick people. I think the other very significant point you made is that the health of the soil is directly related to the health of our gut. And, that for me was a very, very important and telling statement. I fully get it. I fully get it. You talked about the fact that the soil is a living ecosystem, and anyone who's done even a little bit of gardening, even in a pot in a balcony, would certainly know that it is a living ecosystem. You talked about the fact that there is an interlinkage between the physical, the chemical, and biological aspects of putting all of this together. I was very intrigued to learn that there are 6 SDGs that are directly impacted by the whole idea of creating healthy soil. Indeed, that's true. And it tells us how closely interlinked all our various systems are, and why we need to look holistically at the whole environment that we are a part of. The concept of One Health, the health of the biome, the health of animals and everything else around us being one, and therefore the need to focus on what is primary to make sure that all of this does remain healthy. Today's focus on the rhizosphere, that's the root and soil, and how that's an important determinant of the health of the plant. What engineering can be done to improve the quality of the rhizosphere? How does this whole nexus between the plant, the microbiome, and the soil work? And the work that Dr. Sharma and her team have been involved in, she talked about 4 specific areas: hyperspectral imaging, the farmer-friendly strategies to improve soil in brief. But then we looked at this whole, the basic work in a lot more detail. I wish you'd been able to talk to us in more detail about many more of these aspects, and I hope you will do that in the coming months. But after listening to this, I understand the importance of microbes. We all do home composting, right? And they give us a little packet that we are supposed to add, and we don't understand why we're doing it. We do it because they tell us to. But this talk has put that whole thing in perspective and said, this is why it's important to do it, and why we should do it, and how it all fits in together. That was really nice. Just taking off from the research that Dr. Shilpi presented, the combination formulation was the one that gave the best results. She talked about how thereafter the research moved into the field and was actually applied in a farmer's field, and the kind of results that it has shown. And I found another thing very interesting, which is that, and this is for lay people like us who don't really understand the importance of the work that you're doing, which is that plants actually select the microbiome that they are in, and they select the bacterial and fungal composition and so on, which help them combat the diseases and help them become more productive. Much like we do, it's exactly the way our bodies do as well. It's very nice. You use a scientific name, but basically it's tur dal and moong dal, two dals that are very important to all of us across the country, and how the work that you've done has impacted both in terms of productivity and the health of the plants. There is a lot of work remaining to be done. So especially for aspiring folks who are or who want to pick up and do work in this area, climate change, drought, and how does using this rhizosphere-based methods of microbiome engineering actually work? What is it going to help us with? More and more important as we get into these frequent climate incidents that we are seeing happen around us. And the second question, can it be really done at scale, which is really what we would need, so that a nation that preserves its soil preserves itself, just paraphrasing where you started from. So thank you ever so much, Dr. Shilpi, for this absolutely fascinating discussion and chat. Dr. Meenakshi, your work itself is also in the same space, and you've really enriched this conversation today by bringing in a lot of discussion, very rich discussion around all of that. To our audience, thank you for being here. Thank you for your questions. It's always the questions that enrich the discussion, so thank you for that. We will be uploading a recording of this whole talk into the IITACB YouTube channel, and it will be available for viewing. Please feel free to share it with anyone, absolutely anyone, everywhere, anywhere, because what we want to do as IITACB is really to inform people about what is happening in the area of science and technology, and really, make everyone feel comfortable with all these developments around them. So thank you once again, and Jai Hind.
Thank you.